Welcome back to another AP Biology video. This is the second in our series on the processes involved in the central dogma of molecular biology. In the previous video, we talked about how we're able to make a readable, usable copy of mRNA from a DNA transcript through the process of transcription. And today we're going to talk about how we take that molecule of mRNA and use to create a string of amino acids to form a protein. So if you guys are ready, let's jump into it. Now, we discussed in a previous lecture the differences between RNA and DNA. When it comes to RNA, RNA can be bound into a number of different versions or structures that are all based on the type of function that they perform within the cell. Very similar to how different proteins can form different structures in order to match with their functions. Three of the types of RNA that we are going to be primarily focused on include messenger RNA, ribosomal RNA, and transfer RNA. Each of these can be shortened to just the first letter of what their function is and RNA. So we have mRNA, rRNA, and tRNA. Starting with messenger, this is the message. That is the information that was encoded from DNA. This is your usable copy. Ribosomal RNA forms part of the structure of ribosomes in conjunction with some other proteins. And then transfer RNA is going to carry amino acids from the cytoplasm to the ribosome and allow the formation of that amino acid sequence during the process of translation. So when we go to move from each of these biochemical languages, we're going to follow this essential three-step process. The first is that we're going to use the coding strand of our DNA to reveal our mRNA sequence. So this was essentially what happened during the process of transcription. Next, we are going to segment out our RNA sequence into three nucleotide segments called codons. Each codon is going to code for a particular amino acid or function within the process of translation. So at the third step, once we have identified our codons, we can identify the amino acids that correspond with each of these codons and build our polypeptide sequence. Now, this is not something that I need you to be able to memorize because we have a tool that allows us to determine which amino acid is associated with each of our codons. And we use this codon chart, which comes in a variety of different forms, but this is the form that they use on the AP exam. So you'll notice that each of our rows is divided into our first bases, so that's the first letter in our codon. Each of our columns is separated into the second base, and then each row is subdivided into four smaller rows, which represents our third base, or the third letter in our codon. So let's take a look at one of these codons that we have here in our sequence. Let's say we wanted to figure out which amino acid would code for the sequence AUG. So what we're going to do is we're going to find each of our letters. We're going to go A for our first base, so we know that it's in this row. We have U for our second base, so we know that it's in the first column. And then G for our third base, so that's the fourth sub-row in that row that we've already identified. Wherever all three of these items overlap, that is going to be our amino acid. So we can see here that this codes not only for the amino acid methionine, but also as a start codon. So this is going to be the beginning of our translation process. We can do this for all of our amino acids. So if we have the base CGU, we'll see that that codes for our amino acid, which is arginine. We have next, let's clear off some of that and go CAU. And here we have histidine. So here where we have the overlap of all three. We'll go GAG. So let's go GAG. We've got not leucine. I'll fix that in post, but that should say glutamine. So if you're actually watching this video, it will be fixed by the time that I make it. But when I am recording, it is incorrect. And let's clear that off and do our last one. Let's go UAG. That is a stop codon. So our stop codon is where we terminate or end the process of translation, and we have our fully formed polypeptide. When it comes to translating proteins mRNA sequence, we have a couple of key players that we're going to want to keep in mind. The first of these is the ribosome. You may remember ribosomes from when we talked about parts of a cell. These are our structures that are the sites of protein synthesis. They perform transcription, and they are where we're going to build our amino acid sequences. And then one of our other types of RNA, tRNA, are going to carry our amino acids, and these contain what we call anticodons that are complementary to the codons on our mRNA, and that is going to be how they pair up with the correct codon. When it comes to ribosomes are composed of units. We have a small subunit and a large subunit. And each of these subunits is composed of different amounts of RNA, rRNA, and proteins. And you can see here that the components of our ribosomes, when we look at our prokaryotic cells and our eukaryotic cells, are slightly different. This is one of the ways that we're able to distinguish between these two types of cells. And an interesting fact is that the prokaryotic ribosomes, those 70S, which are smaller than the 80S ribosomes that we find within our eukaryotic cells, are the ones that are found within our believed to be endosymbiotically acquired organelles like mitochondria and chloroplasts. Now, like every other part of this unit, what we are going to do first is we are going to go through the process from start to finish. So if you guys are ready, let's start at step 1. First, our small ribosomal subunit is going to bind to what we call a recognition sequence. This is our start codon. AUG is going to be the beginning of all of our polypeptides. From there, we are going to combine with a large ribosomal subunit. This forms what we call an initiation complex. And we're going to have a methionine charged tRNA occupy our P site. So this is the site where we are going to be building our polypeptide strand. So from here, we have two open sites. We have an E site and we have an A site. So you can think of the A site as our accepting site because this is get new amino acids to be pulled in. So incoming tRNAs bind to the A site, and these amino acids will be linked with the previous by an enzyme known as peptidyl transferase. And this is found within or as a component of that large ribosomal subunit. And we are then going to transition over to the E site. We're going to shift our codons over, and we are going to continue to add new tRNAs as new codons reach that A site. Now, this will continue, and our polypeptide strand will continue to grow until we what is known as a release factor, which is coded for by our stop codons. So once we reach a release factor, It is going to disconnect all of our ribosome components and our tRNAs and our RNAs, and we are going to be left with the product of this whole gene expression process, our polypeptide. Now, over the course of this and the previous video, we have discussed different components that help us to distinguish prokaryotic cells from eukaryotic cells. First off, when we compare the gene structure of these two types of cells, we can recognize that prokaryotes don't need any additional processing once we create those mRNA products. So we have very continuous coding regions. There are no interruptions, whereas eukaryotic cells are discontinuous. They contain those interrupting sequences or introns that have to be taken out before we can splice everything back together. Transcription in prokaryotic cells is going to take place within the cytoplasm because we lack a nucleus, so our DNA is free floating. And because we are happening in the cytoplasm, we are often able to do transcription and translation at the same time. Eukaryotic cells, where we've got our nuclear membrane separating our DNA sequence from the ribosomes in our cytoplasm, they're going to keep these two processes separate. They have to take place where they're getting their information from. We have absolutely no post-transcriptional modification in prokaryotic cells because they have continuous regions of code. They are often going to do translation at the same time that they do transcription. So there's really no time and no need for us to really mess around with any of these sequences. Eukaryotic cells, on the other hand, we have to take the time to cut out the pieces that we don't need involved in splicing. And then we're going to add on those additional pieces like the 5' cap and the poly A tail that help to move us out of the nucleus and into the cytoplasm. The ribosome structure is something that we introduced here today, in that the prokaryotic ribosome is slightly smaller than our eukaryotic ribosome, and this helps us to distinguish between these two types of cells and where different components may come from. And finally, our main focus of our video today was on translation, which in both prokaryotic and eukaryotic cells occurs within the cytoplasm, where we find all of our ribosomes, but it's whether or not we accomplish this step at the same time as transcription in prokaryotes, or separate once we've done all of those post-transcriptional modifications in our eukaryotic cells. In the previous video, this central dogma, this flow of genetic information where we go from DNA to RNA to protein. And we talked about how this is mostly unidirectional, except for that communication between DNA and RNA, where we can actually undergo what we call reverse transcription and go from RNA to DNA. So why in the world would we want to do this? Well, there are things that exist that like to attack our cells, like viruses, that have genomes that are composed of RNA rather than DNA. We also probably recognize that viruses are not able to replicate themselves without a host. So when we have a virus, and in this case, we're using a retrovirus, which is specifically used because it has that RNA genome and hijacks cells for their ability to create proteins. And they use an enzyme that is known as reverse transcriptase. Reverse transcriptase takes the RNA from the virus and converts it to a DNA copy, which it can then integrate into the genome of the host cell and get the cell to do all of the work for the virus. So this is one of our examples of the central dogma working in a little bit of reverse, but remember, everything else goes in the correct direction. It's only when you have specialized features that allow you to do other things. So in the final video of this selection, we are going to look at what happens when things go wrong. I'll catch you in the next video, guys. Bye.